Okay, hi everybody. My name is Toby Hocking, and I'm very excited to present my recent work on the generalized functional pruning optimal partitioning algorithm. That's GFPOP for peak detection in large genomic data. And so this is ongoing work, already published in the PeakSegDisk R package on CRAN, and we have an archive preprint available that describes all of the math. It's currently under review at JSS, and we just got the reviews back, so we'll be doing revisions pretty soon. So if you have any feedback, please, I'd be more than happy to talk to you after the talk about that. And so yeah, this is joint work with my collaborators Guillaume Rigaille, Guillaume Bourque, and Paul Fernhead. So in this talk, we're going to be discussing an algorithm for analyzing chip-seq data, which I'm displaying in this plot here. So you can see that there's 4 different rows with these gray bars going from left to right. So the x-axis is positions along the genome in this plot. And the higher gray bars represent more likelihood of binding in this particular chip-seq assay. So essentially these data are interpreted in terms of peaks and background, peaks being where there is a significant binding in this chip-seq assay, and background being where there's just kind of background noise and no significant proteins bound at that part of the genome. So yeah, the typical interpretation is in terms of these binary classification models that they call peak detectors, which I'm showing as like a black bar in this plot. So essentially what this amounts to doing is for every sample and for every position of the genome, you have to classify at that position whether or not there's a peak. And so the peaks are the larger values and the background are the smaller values. So there's many heuristic algorithms that have been proposed for this task. One of them is being shown in black here. This is the MAX2 algorithm. But typical heuristic algorithms suffer from quite a lot of false positives. And so that's going to be the main thing that we address in this talk. We're going to propose a concrete statistical framework to address this issue, and we're going to solve the problem exactly. So the framework that we use is maximum likelihood change point detection with up-down constraints on adjacent segment means. So in the diagram, you can see what one of the chip-seq profiles would look like. The mathematical notation that we use here is Z1 to Zn for the different data points along the genomic position axis. And then in this kind of model, there's 2 different kinds of optimization variables or model parameters that we have to optimize. So on the one hand, we have to optimize these change point variables T1 and T2 in this picture, which can go left and right in order to optimize where the peak positions are. And then on the other hand, we have these segment mean variables U1, U2, and U3 that can go up and down to get the right segment mean for that particular part of the genome. And so what we do is we fit this variable number of segments to a particular genome subset by solving this minimization problem, which is equivalent to a maximum likelihood problem. So the loss here that we use is Poisson loss because we have count data. And so what this objective function says is that we just are summing over all segments from k equals 1 to s. S is the number of segments. And then for all data points, I on each segment. And so this is the really important thing of our contribution. We consider these inequality constraints subject to mu k minus 1 less than mu k for all even k, and subject to mu k minus 1 greater than or equal to mu k for all k which are odd. And so these constraints enforce a segmentation model which changes up and then changes down and then changes back up again. So it's very easy to interpret in terms of peak and background, right? So the first segment and all of the odd number segments are background, and then the second segment and all the even number segments are peaks. And so naively, this is an integer programming problem, and it's quite difficult to solve with off-the-shelf solvers. And actually just enumerating all the different possibilities would result in an exponential time algorithm. And so actually one of the main contributions is figure out an efficient way to solve this problem. And so so that's what we described previously at ICML in 2015, which is this quadratic time approximate algorithm for solving this problem. And then previously we also described this algorithm which is linear in the number of segments and log linear in the number of data points for solving this problem. And so again, to emphasize the importance of the up-down constraints, I've I've included this slide where here I include the unconstrained segmentation model. So this is what the model looks like when you don't include these constraints in red here. But if you do include the constraints, you get this nice model which is easily interpretable in terms of changes up to peaks and then changes back down to background, followed by changes up back to peaks. So you don't always get that with this unconstrained model, which can change up and then up again and then down again, depending on the data. And in that case, it's not directly interpretable in terms of peaks and background. So what we propose in this most recent paper is this penalized version of the previous problem. So this is what we solve with the generalized functional pruning optimal partitioning algorithm, or GFPOP. So instead of having this S parameter for the number of segments, we have this lambda parameter, which is a penalty that controls the number of segments. And so we get a similar optimization problem with similar constraints here that enforce either increasing or decreasing changes. But here the objective function now contains not only the loss, but also a penalty. And here the penalty here includes this indicator variable, which counts the number of changes. And so it's an optimization problem, which includes variables for the segment mean, the hidden states, and the change point variables can actually be solved by our algorithm in log linear time in the number of data points. So that's n log n. And so actually there's a nice interpretation of this model in terms of a hidden Markov model, or this graph here, where we have these two states, S equals 0 for background, S equals 1 for peak. And so at every segment, you're either in the background state or the peak state, and the edges in this graph represent the possible change point variables that you can use to go between the states. And so actually this algorithm is quite a lot more general, and it can be applied to any possible graphs like this, so that's what we're pursuing in future work. But yeah, to get to some results, here's some figures that show you how fast our algorithm is, right? So I said that it's n log n, and so on the left figure here, I'm showing you how many candidate change points the algorithm is storing for different data set sizes. And you can see that even going up to data set size with 10 million data points, you see that we only have to store, on average, 19 intervals or candidate change points. And so since the algorithm is linear in the number of these intervals or candidate change points, that means it's very scalable to large data. So in fact, we see that it scales as log n with the number of data points. And overall, I did some timings and some measurements of the space that this algorithm takes. And again, so we see that it's log linear in the number of data. So even for really large genomic data sets, subsets like 10 million data points, it only takes on the order of hours and tens of gigabytes, which is actually quite manageable. And so yeah, just to give you like a real world example of what the numbers would look like, again, for 10 million data points and 1,000 peaks, the previous algorithm would require about 100 terabytes of storage space and 12 weeks of computation time. But with this new algorithm and the right penalty that would get 1,000 peaks, the proposed algorithm only takes about 1 hour and 80 gigabytes of data storage. And so to actually use this stuff in R, what you do first is you write your data set to a bed graph file. This is kind of a standard file in the bioinformatics community. It's just a 4-column CSV file, basically, that has the positions and the data values. And so after you write it to the disk, then you call our solver, which is implemented in the PeakSegDisk package, where you tell the path to the folder that contains this coverage.bedgraph file, and then you tell it what penalty you want to use. Then it runs the optimal solver, solves this optimization problem. It gives you this fit object. One of the components is a loss that tells you various things about the model, like what was the optimal loss value and how many peaks ended up for that particular penalty. And we'll save it to the end. And another thing that you can do is you can do some visualization of the model in this fit object. So here I'm doing a ggplot that shows the data again in gray in the background here, and then I'm using a geom segment with fit $segments to plot what the segmentation model looks like. And so in this case, this model looks good, like it's capturing the peaks in this data subset. And so kind of to quantify how this model is working on a larger scale, what we use is this procedure that we described in Bioinformatics in 2017 with these manually labeled regions in different genome subsets. So we had a biologist manually go through and label some data and so the biologist gave us these regions that say in the pink regions there should be a peak start, and in the red regions there should be a peak end. And so what we do is we use those regions to quantify how good our model peak predictions are. So for example, when the penalty is very large, we get a model with too few peaks, like there should be a peak here in this pink and red region, but there's not, so we call that a false negative. And with a small penalty like 6,000 here, we get a model with too many peaks. So that's resulting in what we call false positives, because actually in these pink regions, for example, there should only be one peak start, but here there's more than one, so that's also bad. But by adjusting the different penalty values, you can see that there's a range of penalties here between about 9,500 and 267,000 that result in no errors with respect to the labels. And so that's between 34 and 236 peaks in this data subset. And all of these models achieve 0 label errors. So this is the kind of protocol that we were using to evaluate the accuracy of the different methods. And so what we do is we're training our model by using this kind of supervised penalty learning strategy that we've previously described at ICML in 2013. I'm not going to go into all the details right now, but essentially we're using the labels on some parts of the data to learn what the lambda values should be and then predict the right number of peaks on the other data sets that we want to make the predictions on. And so the results that we presented in this archive paper in 2017 show that when we do this comparison of these different algorithms on the y-axis in terms of test AUC, which I'm showing you on the x-axis, for all of these 7 different data sets, which I'm showing you in the different panels, well, what we see is that our proposed algorithm here at the top is doing quite a lot of better on all of the data sets than the previous baseline. So here I'm showing you 2 baselines, MAX and HMCAN broad, which are heuristics from the bioinformatics literature, which do not do this formal solving of an optimization problem, and they have lots of tuning parameters which may be suboptimal for the different data sets. And so you can see that some of them are good for some data sets, but not for others. For example, MAX, it's not so good for the broad data sets, these first 3 panels here, but it's quite good for it's doing okay for the other data sets. And for example, the HMCAN broad data algorithm is doing okay for the broad data sets in the first 3 panels, but not so good for these other data sets in the last panels. Whereas our algorithm is doing quite well in all of the different kinds of data. So this kind of highlights the power of mathematical optimization combined with machine learning. It's really able to learn how to call peaks, do this structured binary classification problem in all of these different kinds of data sets. And so yeah, just to conclude, I'll just summarize by saying, again, our proposed GFPOP algorithm computes the global minimum of this hard, non-convex, constrained change point problem in log linear time. It only takes log linear disk space and only log memory. So you can do this on a standard laptop, no problem. And so the peak predictions, like I showed on the last slide, are highly accurate in labeled genomic data sets with 2 different kinds of patterns, unlike some of the previous baseline algorithms. And we have, again, this PeakSegDisk R package, which implements this algorithm in C++ code with a nice R interface. So please contact me at Toby Hocking at nau.edu if you have any comments, or please talk to me after this.
So thank you for this nice presentation. So do we have a question?
Yeah, we had a question from Dirk back here. Okay.
Hi, thank you. Do you have any plan to take into account chip negative controls that are sometimes called inputs?
Yeah, sure. So there was a question about if we have any way to take into account the negative controls or input samples that are typically run when we do chip sake analysis. And so, yeah, with this particular model, there's no direct way to take that into account, but we have another algorithm in a previous archive paper, I think 2015, that is doing a joint detection across multiple samples. So then you can kind of see if there are peaks in the experimental sample and not in the corresponding input sample. So you might want to take a look at that.
So any other question?
Sorry. Do you know what IC data is? Chromatin conformation capture. Do you think you can apply such algorithm to detect IC contacts, so that are like two-dimensional peaks?
Yeah, so there was some question about the high-C data, which is a two-dimensional data rather than the one-dimensional data that I showed in this talk. And yeah, there have been some previous papers about using change point detection approaches to those data. So it wasn't by our group, but there are some people looking into that, so you might want to do a search.
Any other question?
Hi, thanks for a nice talk. I was just curious, when you decrease the size of the penalty, does it tend to just cut up peaks, or do you tend to discover new peaks? It's sort of like from the small example, it looked like it seemed to be breaking up wider peaks more. I was just curious if in practice you had any.
Yeah, so the question is about what happens when you change the penalty. Is it making peaks bigger or smaller? Is it breaking them up? Or is it hierarchical? And so, yeah, unlike the binary segmentation heuristic, which will start with a model and then add or subtract change points or peaks, that's not what we do. We solve this problem optimally. So we don't know in general if, by decreasing or increasing the peak, it's going to have any peaks that are the same or different than that previous model. It's just the result of solving the optimization problem, so we don't know necessarily. It might be something that you could prove, but we haven't looked into that.
Hi, nice talk. First of all, what do you have for a measure of enrichment for your peaks? Because for example, Max outputs nicely, it's for this measure of enrichment. How do you just call the peaks in a binary way, or you also have a measure for that?
So the question is there a quantitation of how big the peak is? And so, yeah, we can just use the mean of the region. So you can see there's some peaks which are bigger, and that results in a higher segment mean here, where there's some peaks which are smaller, and that results in a lower segment mean.